Thanks everyone for joining me today. I'm very excited to share our work with you on the de novo design of G-protein mimetics. And thanks very much to Twist for setting this up and for inviting me to speak. So today I'll tell you about what G-protein mimetics are, why we need them, what we can use them for. Then I'll describe the computational de novo design of G-protein mimetics. How do we make these things? And then I'll wrap up with a description of our production and testing in the wet lab after we've designed these things in the computer. So first, just a brief introduction to G-protein coupled receptors. So GPCRs are really the most important family of signaling proteins that we have. So they're integral membrane proteins. They have 7 transmembrane domain spanning regions. And they mediate a myriad of critical signaling processes in the body. So GPCRs couple to G proteins. These are heterotrimeric signaling molecules. There's a variety of different subtypes, and they plug into all the usual suspects, AKT, NF-kappa B, MAPK, you name it. So our genome encodes about 800 GPCRs. A little over half of these we use for seeing, smelling, and tasting, and the other half are the drug targets. And so these recognize everything from hormones, neurotransmitters, pH, lipids, nucleotides, mechanical force, peptides, lots of really important stuff. And critically, over a third of all the drugs we have approved by the FDA act by targeting GPCRs. So it's an incredibly rich area for drug development. And biologics are also a growing facet of the drug development. So a few years ago, the first monoclonal antibody targeting a GPCR was approved. And as you can see, there's a lot more biologics that are currently in development. So this is very much a frontier. So how do GPCRs work? So GPCRs transmit information through changes in protein structure. So here we've got a structure of the GPCR in the inactive state, where it's not signaling. And when it binds a ligand in the extracellular surface of the receptor, it changes its conformation. And this conformational change propagates across the membrane to the interior of the cell. And so you can see that both the exterior surface are in different conformations between these two states, and the interior surface of the GPCRs are in different conformations between these two states. And ideally, what we would like to be able to do is to control the conformational dynamics of these GPCRs and stabilize them in the inactive or active states at will. So a G-protein mimetic mimics the endogenous binding activity of a G protein and binds to the intracellular surface of a GPCR. And there's lots of useful things you can do with a G protein mimetic. Proteins that are conformationally dynamic tend to be less stable. And so freezing out some conformational flexibility in a receptor can be used to make it more stable and make it easier to express and purify. This is often done with small molecule ligands. And the ability to do this with a protein ligand is also really advantageous. While there are many structures of GPCRs, determining structures of GPCRs in the active state remains a challenge. Oftentimes, this is done using the full heterotrimeric G protein complex, oftentimes with various stabilizing immunoglobulins. So it's still a fairly laborious and delicate process to do. Also, engineering antibodies that are conformationally selective, binding to the extracellular surface, remains a challenge. You need to be able to have, ideally, the ligand, the target protein, stabilized in both states, active and inactive, in order for those selection campaigns to be efficient. And finally, you can also develop novel GPCR signaling assays if you've got proteins that can bind and recognize the intracellular surface and distinguish between conformations there. And this has applications in both development of novel GPCR ligands, as well as trying to leverage GPCRs for synthetic biology. So our goal in this project here is to design G protein mimetics that work with all human GPCRs, not a GPCR, but sort of a pan GPCR solution. So, we didn't invent G protein mimetics. This concept has been around for a while. And there's really two prevailing technologies. The first one is nanobodies. So these are single chain IG domains from camels and alpacas. And nanobodies are really great, and it's possible to make them bind selectively to the active state of a GPCR at the intracellular surface. And they can be really high affinity, and they also tend to be highly specific to a given receptor. And so this is great if you want to be able to have the precision to dissect the mechanism of a specific GPCR. But if you want to have a single platform technology that you can apply to many different GPCRs, this becomes problematic. It's also incredibly challenging to raise these nanobodies. There's often a unique discovery campaign that has to be performed using an existing small molecule ligand in order to find the nanobody that binds to the intracellular surface. So it's not a very general tool if you wanted to, say, create a mimetic for an orphan receptor where you don't have a known ligand. Nanobodies also have a disulfide bond. Some of them can fold under reducing conditions in the interior of a cell, but not all of them. And so we want something that's also ideally able to tolerate a wide variety of redox conditions. Another type of G protein mimetic is this protein called mini-G. It was developed by Chris Tate's group at the MRC. And mini-G is a really fantastic protein. It was engineered starting from the GS subtype, a human G protein alpha subunit. And some of this protein was chopped off and some new linkers were attached. And it's really quite remarkable, and it works very well. But it's not a one-size-fits-all solution. So mini-G itself, the structure is fairly detergent sensitive, so it's denatured in the presence of detergent. It does require reducing conditions. There's some unpaired cysteine residues, and it tends to ladder if you don't have reducing. If you don't have a reductant in the solution. And GPCRs also have disulfide bonds in the extracellular loops. And so it does require some very careful tuning of the redox conditions to use mini-G. Importantly, the mini-G concept is not extensible to all G protein subtypes. So it's not possible to create a mini-G for all the different types of GS. And so therefore, we can't make a mimetic in this way for all types of human GPCRs. So we really want to make an ideal G protein mimetic that's widely extensible. And so we think we can learn a lesson from mini-G and then apply de novo protein design to really create an ideal one. So if we look closely at the structure here, we have looking at the structure of the beta-2 adrenergic receptor bound to the GS heterotrimer. So the GPCR is in gray and the G protein is in orange. And a component of the G protein I've highlighted here in magenta. This is the alpha-5 helix. It's at the C terminus of the alpha subunit. And this one helix, if we zoom in here, contains over 70% of all the atoms in the G protein that contact the GPCR. And so this is the primary specificity determining region of a G protein. And it's also contains the majority of the interactions. So we think this is a great place to start if we want to build an ideal G protein mimetic. It's also highly conserved. So we've got the GS heterotrimer here on the left, and on the right, here's a structure of a different GPCR, the mu opioid receptor, bound to the GI heterotrimer. And you can see the alpha-5 helix in magenta here is highlighted in both cases. It's present essentially in all the G protein GPCR interfaces. So this does seem to be like a general structural feature that we can use and exploit for a wide array of mimetics. So this is essentially what we want to do. We're going to take this helix out of the G protein, throw away the rest of this complicated molecule that does very important things in the cell. But for our purposes, we don't really want. And we're going to take this helix and we're going to wrap a new protein around it that will pre-organize this structural component so that it can bind to the GPCR. So how do we build proteins totally from scratch? So we use computational design, and computational design is unique in that it lets you explore regions of protein sequence space that are otherwise inaccessible to natural selection. So if you think about it, proteins are related to each other evolutionarily, right? The way you get one protein is that it evolves from another through a sequential series of changes. This means that the proteins that we find in the organisms in the genomes of organisms are not a random sampling of amino acid space, but they're clustered, where we see related proteins that are similar to one another clustering together. If we think about the totality of the space that's accessible to protein as a material, it's really, to say it's galactically huge is actually a rather gross underestimate. So if you look in UniProt today, the average protein is 336 amino acids long. So nothing extravagant, just average protein length. So that means that the search space available to the average protein is 20 to the 336 power. Currently, our best estimates for the number of atoms in the observable universe is about 10 to the 80th. So we have explored a fantastically small portion of the potential that protein has as a material. And evolution has also sampled a fantastically small fraction, and it tends to be sampling things that are related to one another. Directed evolution can push back the bounds that you can sample, but still around regions of sequence space that are similar to things that have already existed. In de novo protein design, we're not bound by that, and we can go deep into these unexplored regions of sequence space using computational methods. So the protein design world really came out of the protein structure prediction world. And in structure prediction, we start with an amino acid sequence, and then we use this sequence to try to predict what the tertiary structure of the protein looks like. I'm sure many of you have heard the news with AlphaFold 2 recently, largely solving this problem for monomeric proteins that have enough known homologous sequences. Protein design is really this process run in reverse. So if you have a backbone with no amino acids on it, we can put amino acids in each position in that backbone and come out with our primary amino acid sequence. And many of the same tools that were originally developed to do the structure prediction actually are applicable for doing the design as well. And so we use Rosetta software to do this as our primary tool. But how do we get this backbone? And so for this, we use a method called sewing, originally developed in Brian Kuhlman's lab at UNC, and that we've extended here at the Institute for Protein Innovation in Boston. And so sewing works by taking fragments of known protein structure and aligning them together to create new topologies that are unlike anything that has existed before. And once you have your backbone, then we can perform our sequence design step. And importantly, we can stitch these fragments of protein structure together in context. So for instance, if we had a single alpha helix in context bound to a GPCR, we can wrap a new protein around that single helix to turn it into a full protein. And that's exactly it. So here I'm showing you the beta-2 adrenergic receptor again with the GS heterotrimer bound. Here's our de novo mimetic. And so our de novo structure is shown in cyan. You can see we still retain this starting helical fragment from our, and built a new protein around it. So just an example of what a couple of these structures look like. So we designed a suite of these in the computer, and then we go and get synthetic genes for these and produce and test them in the lab. And sewing is really great because it creates lots of different structures that satisfy your functional requirements. You don't have to define a topology or what you want the protein to look like ahead of time. You simply say what you want it to do, and the algorithm will create a new topology that is capable of doing that. So here's our starting fragment, and you can see the different shapes that come out of them are very different. And if we color this from the beginning, the N terminus to the end, the C terminus, in a rainbow color from blue to red, you can see here that the structures are quite different. The beginning of it starts on different sides of the structure, even though the end is all conserved. To sort of drive this home, we produced and tested 48 designs in the laboratory, and I've aligned all of them here by that fragment we used to design them. And essentially what you see here is a bird's nest. We're not getting any real structural conservation outside of the starting fragment. Everything is really different from each other, but they all manage to stabilize this helix in a significant way. And if we align the sequences of these 48 designs, you can see here that the C-terminal region is highly conserved, and everything else upstream of it there's no conservation. Everything is really quite different from each other. If we try to build an evolutionary tree, we get a starfish. So these things are totally unrelated to each other. And if you blast them against known sequences, the only thing that pops up with any sort of identity to anything extant is the C-terminal helix that we used to design. And the rest of the sequence is completely unrelated to anything seen in nature before. Okay, so now we're going to shift gears from in silico and now move in vitro and produce and test these. So we express and purify them all in the laboratory. Remarkably, we're able to obtain soluble material for the vast majority of our designs. So about 90% of them can actually be purified. This is, I would say, showing that de novo design is becoming a more mature technology. This is traditionally much lower hit rate. So we're getting pretty good at making proteins from scratch nowadays. And we wanted to test to see if they actually are capable of binding to the GPCRs. So to do this, we used a pull-down assay. So we purify our G protein mimetics, and we attached a biotin molecule to them. And then we can use this to couple the mimetics to streptavidin beads. We then take our GPCR, and the GPCR we purify in detergent micelles, and the GPCR has a flag tag on it so that we can identify it. We can treat the GPCR with an agonist that should lock it into the active state. And if our G protein mimetic is capable of binding to the GPCR, we should get receptor pull down, which we can detect by Western blot. If we take our GPCR and we treat it with a small molecule antagonist that should lock it into the inactive state, if our G protein mimetic is able to distinguish between different conformations of the GPCRs, that is to say, if the binding is conformationally selective, then antagonist treatment should block the association between the mimetic and the GPCR, and we should not see pull down. So I won't show you the full suite of 48 mimetics, but I will show just a couple here. So we tested this with adenosine 2A receptor as well as the beta-2 adrenergic receptor. A2A receptor is what we used for the structure for the design, and beta-2 is an unrelated receptor to test the generalizability. Our negative control is just another random protein that shouldn't bind. Our positive control, in this case, we're using the mini G. And we can see that we have pull down in the presence of an agonist for both receptors and not in the presence of an antagonist. And then 3 of our de novo GS mimetics, these are design number 13, 18, and 23. These are just arbitrary numbers. They don't relate to the design quality, just the order in which they appeared in our gene synthesis. You can see we're able to get conformationally selective pull down, where we are able to get pull down in the presence of agonist and not in the presence of antagonist. Beta-2 is interesting in that we're able to get some selection in that we're getting greater pull down in the presence of the agonist, but we're still seeing a little bit of weak pull down in the presence of the antagonist. We're looking into why we're seeing this. And just a reminder that these structures are all alpha-helical. And when we do circular dichroism spectroscopy, which reports on the secondary structure content of a protein, we see a classic alpha helical signature, which is essentially two negative peaks, one at 208 and another peak at 222 nanometers. And so this is the classic signature that our protein is alpha helical. And when we heat this protein up to 95 degrees, essentially it flexes just a teeny bit. And when we cool it back down, it returns. And here we're just monitoring some of the peaks here, the alpha helical peaks. And this would be if there was beta sheet, this would be a beta peak. We get a flat line in the melting curve as we increase the temperature. So this protein doesn't melt. It's essentially a brick. This one is for the de novo G27, but this is the same curve we see for all of our G protein mimetics. So our designs are hyperthermal stable, and they appear to be well folded, adopting helical conformations. So this story is a work in progress. So this is where we're at right now. At the moment, we're working to genetically fuse our G protein mimetics to the GPCRs with a cleavable linker so that we can really lock the receptor in the active state by forcing its association with the G protein mimetic, essentially forcing an inside-out conformational change. We'd like to use this as an antigen in an antibody discovery campaign to try to find conformationally selective antibodies that recognize the extracellular surface of the receptor. And we're also working to convert our GS mimetics into G protein mimetics for other subtypes, I, Q, and 12. So I'd like to thank all the members of my group for really just this fantastic group of folks to work with. In particular, Ian Trubridge and James Bowman and Isabel Mujigemba have done a lot of the work on the G protein mimetic project, and Ian actually produced all of the data that I shared today. We've had a fantastic collaboration with Andrew Cruz at Harvard, as well as with Brian Coleman at UNC. Thanks to our funding, and thank you all for your attention.